Light filters down through the surface of the water, breaking into shifting patterns across the reef below. Structures rise from the seafloor, branching, layered, alive. Fish dart between the branches, weaving through gaps in the coral as they move through the water. Out beyond the reef, the seafloor falls away, the structure disappears, and with it, anything fixed in place. Here, something drifts, a translucent bell contracts, then relaxes. The body pulses forward, suspended in the open water. One builds, the other drifts. One shapes entire ecosystems that stretches for kilometers. The other moves with the currents and has no fixed shape at all. And yet, these are the same kind of animal. Corals, jellyfish, and even less well-known forms, so small they are invisible to the naked eye. All belong to a single lineage, the Cnidarians. To understand how these forms are related, we need to construct that lineage, step by step, because the diversity we see is not random, it follows a pattern. Before we can understand their differences, we need to ask a simpler question. What is a Cnidarian? Cnidarians are animals in the phylum Cnidaria, a group that includes jellyfish, corals, sea anemones, and hydrozoans, among others. It is worth briefly noting that, despite the name, not all animals commonly called jellyfish belong to this group. Comb jellies, for example, are part of a completely separate lineage. At first glance, Cnidarians appear to have little in common. Corals resemble plants or stone more than animals. Jellyfish seem almost structureless, lacking the features we might expect. No limbs, no obvious head, no centralized brain. But beneath these differences lies a shared architecture, a body plan that is at once simple and remarkably flexible. What truly defines the group is something more specific, specialized stinging cells known as cnidocytes. Each of these microscopic structures can be triggered in an instant, firing a coiled thread that can anchor, entangle, or deliver toxins to prey. Virtually all Cnidarians possess these cells, although in some lineages they are reduced or heavily modified. Throughout the phylum, the shared body plan can take two different forms. One form is attached to the substrate, a cylindrical body extending upwards into the water with tentacles arranged around the mouth. This is known as a polyp. The other is free-swimming, a bell-shaped body that moves by rhythmic contraction, with the mouth and tentacles pointing downward. This is known as a medusa. Polyps and medusae are not different species, but are often linked as different stages of a life cycle. A polyp can produce medusae, which in turn give rise to new polyps. But this pattern is not universal. Some individuals remain in the polyp form throughout their lives. Others are dominated by the medusa stage. And in some, one form has been lost entirely. Coordination within the body is not handled by a centralized brain, but by a diffuse nerve net, spread throughout the tissues. Rather than processing information in a single location, signals pass through this network in multiple directions, allowing the animal to respond to stimuli from all sides. This system is simple but effective. It coordinates the extension of tentacles, movement of the body, and the firing of stinging cells. In the free-swimming medusae, it drives the movements that propel them through the water. In attached polyps, it regulates feeding in response to the surrounding environment. In some groups, this organization extends even further. Individual bodies may not exist in isolation, but as part of larger colonies, formed from repeating units of the same basic body plan. These units, known as zooids, are often specialized. Some are adapted for feeding, others for reproduction or defense. Together, they function as a single, integrated organism, rather than a collection of separate individuals. From this shared foundation, the diversity of Cnidarians begins to take shape. That diversity is not random, but it does reflect the vast span of time over which this group has evolved. The earliest members of Cnidaria appear in the fossil record around 580 million years ago, during the Ediacaran period. At that time, complex life on Earth was only just beginning to emerge. These are among the earliest animal lineages we know. Over hundreds of millions of years, this simple body plan has been reshaped, modified, and diversified, giving rise to the forms we see today. To understand how that diversity is structured, we can begin with the first major division within the group. While most Cnidarians alternate between Medusa and polyp forms, one major lineage does not. In the subphylum Anthozoa, the Medusa stage is absent entirely. They remain as polyps attached to the substrate for their entire lives as adults. This may initially seem limiting. If Anthozoans cannot move, then how do they reproduce, and how do they colonize new areas? The answer lies in their life cycle. Many corals reproduce both sexually and asexually. During sexual reproduction, they release vast numbers of eggs and sperm into the water, where fertilization occurs. The resulting larvae are free swimming. While the adults remain fixed, these larvae can disperse through the water column before settling onto the seafloor and developing into new polyps. Asexual reproduction allows further expansion. Polyps can clone themselves, growing existing colonies, or, if a fragment breaks away, establishing new ones elsewhere. Anthozoa contains, very broadly, the corals and sea anemones. They are predators, armed with tentacles surrounding the mouth. Like other Cnidarians, Anthozoans are radially symmetrical, but within this group, that symmetry takes on more specific forms, patterns that define their major lineages. The first major division here is the Octocoralia. They show 8-fold radial symmetry. That is, the same pattern is rotated 8 times around the center of the body. This is extremely rare in the animal kingdom, being a much more common concept in geometry and mathematics. Octocorals, sometimes called soft corals, have 8 tentacles per polyp. There are over 3,000 species, all found in marine or brackish waters around the world. Most do not exist as solitary animals. Instead, they form colonies, networks of connected polyps embedded within a shared structure. These colonies can take on a wide range of forms, from soft, flexible masses to branching fans that extend into the current. Rather than building rigid frameworks, they often remain supported by internal elements, allowing movement with the surrounding water. This represents one pathway, a stable, repeated body plan, multiplied and extended through colonial growth. The remaining Anthozoans follow a different trajectory. In the next lineage, the body is organized not in 8s, but in multiples of 6. Much more common in nature, 6-fold radial symmetry is seen in snowflakes, honeycombs, and certain flowers, and, of course, in Hexacoralia, the next branch on our family tree, also known as the Hexacorals. Some of the most iconic Cnidarians are Hexacorals. Stony corals form vast coral reefs, stretching for kilometers and providing habitat for countless animals. Sea anemones are known for their vibrant flower-like appearances, in close association with clownfish. Mushroom corals look like massive underwater fungus, at least when seen from above. All of these, and more, are hexacorals. Even the diversity within this one group is staggering. Stony corals form colonies, much like octocorals. Sea anemones, however, are solitary, just being a single polyp, and living independently from any others. Many of the colonial hexacorals produce rigid calcium carbonate skeletons. Over time, these structures accumulate, forming the framework that defines coral reefs. A third lineage sits alongside the hexacorals, the Cereantheria, or tube anemones. Historically placed within the hexacorals, they are now often recognized as a distinct lineage, though they remain closely related. These animals retain the familiar polyp form, but live within self-constructed tubes embedded in the substrate. Their bodies are elongated, with tentacles arranged in distinct whorls. While they share the same fundamental polyp structure as other Anthozoans, their development sets them apart. Instead of a short-lived larval stage that quickly settles, they produce a more developed, longer-lived larva that can remain in the water column for extended periods. This allows for wider dispersal, delaying settlement until a suitable environment is found. When they do settle, they construct a tube within the substrate, a protective structure into which the body can fully retract. From this position, the animal then extends its tentacles into the water to feed, while remaining anchored and concealed. Across these lineages, the differences are clear: 8-fold and 6-fold symmetry, solitary and colonial forms, soft bodies and rigid structures. And yet, all remain bound by the same limit. There is no Medusa stage, no transition to a free-swimming form. The polyp is not one stage among many. It is the foundation from which all variation arises. This is one path through Cnidarian evolution, but it is not the only one. In other lineages, the second body form, the Medusa, does not disappear. It becomes central. These animals belong to the Medusazoa, a major branch of the Cnidaria, in which the life cycle is built around the presence of a free-swimming stage. Here, the Medusa is no longer an optional phase. It plays a central role in movement, dispersal, and, in many cases, reproduction. The body plan shifts accordingly. Rather than remaining fixed to the substrate, these animals are able to move through the water column, expanding the range of environments they can occupy. But this transition is not uniform. In some Medusazoans, the life cycle still alternates between polyp and Medusa forms, each stage performing a different role. In others, one stage is reduced, shortened, or lost entirely. The balance between attachment and movement is adjusted in different ways across different lineages. What unites them is not a single structure, but a shared pattern, a life cycle in which the Medusa form is present and often dominant. From this foundation, a wide range of strategies emerge. Some lineages retain a high degree of flexibility, combining colonial growth with alternating life stages. Others are defined by the Medusa itself, larger, more developed, and central to the organism's biology. And, in some, this form becomes more specialized still, incorporating increasingly complex sensory and behavioral systems. To understand how these different strategies arise, we can begin with one of the most flexible and structurally diverse lineages within the group, the Hydrozoa. In many Hydrozoans, the life cycle retains both forms. A polyp produces medusae, which are released into the water column. These medusae then reproduce, giving rise to new polyps. Rather than favoring one stage over the other, the two are linked, each performing a different role within the life cycle. But this is only part of the story. In many species, the polyp stage does not exist as a single individual. Instead, it forms colonies, networks of connected bodies, each derived from the same original organism. These are not simple gatherings of animals. The individual units or zooids are specialized. Some are adapted for feeding, others for reproduction, others for defense. Together, they function as a coordinated whole. In some cases, this division of labor becomes so pronounced that no single zooid is capable of independent survival. The colony behaves not as a group of individuals, but as a single integrated organism, one that is composed of many parts. A striking example of this can be found in the Portuguese man-o'-war. Often mistaken for a jellyfish, it is instead a colonial hydrozoan, composed of multiple specialized zooids. Some form the gas-filled float that keeps it at the surface. Others are responsible for feeding, reproduction, and defense. Individually, these units cannot survive. Together, they function as a single organism, drifting across the ocean's surface. This type of organization allows hydrozoans to explore a wide range of forms and strategies. Some remain small and inconspicuous, attached to the substrate. Others extend into the water column, forming complex drifting structures. Across the group, the balance between polyp and medusa, individuality and integration, is constantly adjusted. This flexibility represents one pathway within the Medusazoans, but in other lineages, the form blurs the line between medusa and polyp. Not all Medusazoans take the flexible approach of Hydrozoans. In one lineage, the medusa form appears, but does not fully separate from the substrate. These are the Staurozoa, sometimes known as stalked jellyfish. At first glance, they resemble small upside-down medusae. Tentacles extend outward, and the body is organized in a familiar radial pattern. But, unlike the free-swimming forms seen elsewhere in the group, these animals remain attached to the seafloor by a stalk. They do not drift through the water column. Instead, they occupy a fixed position, capturing prey as it passes. In this way, they blur the distinction between the two forms. The body resembles a medusa, but the lifestyle remains closer to that of a polyp. This represents another way within the Medusazoans, one in which the medusa form emerges, but mobility does not develop. In other lineages, however, this transition is taken further. The medusa becomes free-swimming, and increasingly central to the organism's biology. These are the Scyphozoa, often referred to as the true jellyfish. Here, the medusa stage dominates. The familiar form, a bell-shaped body, pulsing rhythmically through the water, is not a temporary phase, but the primary expression of the organism. Movement is no longer limited or occasional. It is continuous, driven by repeated contractions of the bell, allowing these animals to navigate the water. This is supported by a structural change. The body of the medusa is dominated by a large gelatinous layer known as the mesoglea. This material provides both buoyancy and support, allowing the animal to maintain its shape while moving through the water. The polyp stage has not disappeared entirely, but it is reduced. Small and often short-lived, it serves primarily as a developmental stage, producing medusae before giving way to the free-swimming form. This shift changes how the animal interacts with its environment. Rather than waiting for prey to pass within reach, jellyfish move through the water, encountering food as they go. Tentacles trail behind the body, armed with stinging cells, capturing and immobilizing prey in open water. Despite this apparent simplicity, the coordination required is significant. The rhythmic pulsing of the bell, the orientation of the body, and the timing of tentacle extension are all controlled by the same diffuse nerve net seen across the Cnidarians, operating without a centralized brain. In this lineage, the medusa is not just present, it defines the organism. This represents a further step along the same trajectory, away from attachment and toward mobility. But even within the medusa, evolution continues to refine and specialize. In one final lineage, this form becomes more structured still, more controlled and more complex. These are the cubozoa, the box jellyfish. At first glance, they resemble other jellyfish, but the structure of the body reveals important differences. The bell is more angular, forming a box-like shape, with tentacles extending from its corners. Movement is more directed, with stronger, more controlled contractions, allowing them not just to drift, but to swim. This shift is accompanied by a change in how these animals interact with the environment. Unlike the largely passive feeding seen in many other Medusazoans, box jellyfish are active hunters. They move through the water with purpose, encountering and capturing prey with greater precision. Their tentacles remain armed with stinging cells, but their use is more targeted, rather than relying on chance contact. This increased control is supported by more developed sensory structures. Clusters of light-sensitive organs allow them to detect changes in their surroundings, orient themselves, and respond to obstacles and potential prey. In some species, this system is particularly potent. The sea wasp, Chironex fleckeri for example, combines strong swimming ability with highly effective venom, allowing it to capture fast-moving prey. It is the largest of the known box jellyfish, and has been responsible for a number of documented human fatalities. Size is not necessarily a good indication of risk, however. The Irukandji jellyfish, Carukia benesi, is tiny, reaching only a few centimeters in height, with just 4 tentacles. Despite this, its sting can cause Irukandji syndrome, a condition characterized by severe pain, anxiety, and nausea that can persist for days or weeks. In rare cases, it can be fatal, and may be difficult to attribute to the animal responsible. Compared to other Cnidarians, this level of effect is noticeable. Stings from many true jellyfish, such as the lion's mane jellyfish or mauve stinger, may cause pain, swelling, and skin irritation. Even the well-known Portuguese man-o'-war, a hydrozoan, not a true jellyfish, can inflict severe welts and pain, but deaths are rare and usually caused by complications like shock, heart attacks, or drowning. Across all these animals, the same basic mechanism is at work, the firing of cnidocytes. Yet in the box jellyfish, the system is deployed with greater precision, and in many cases, with more powerful effects. The underlying body plan has not changed. The same radial symmetry, the same basic organization, and the same diffuse nerve net remains. But within that framework, the level of coordination is markedly increased. Here, the medusa form reaches its most developed expression, capable of directed movement, active predation, and all with the same nerve net used by all Cnidarians. This represents a culmination of this pathway through Cnidarian evolution. But there is one more branch, one that is poorly known, and one that transforms the familiar body plan into something quite different, reduced, simplified, and adapted to an entirely different way of life. These are the Endocnidozoa. At first glance, they do not resemble Cnidarians at all. There are no tentacles, no bell-shaped medusae, no obvious radial symmetry. The familiar forms, the polyp, the medusa, seem to have disappeared entirely. Instead, these organisms are microscopic, living within the tissues of other animals. Many are parasites of fish and aquatic vertebrates, often passing between two different hosts as part of their life cycle. This kind of multi-host strategy is common among parasites, but it represents a stark departure from the free-living lifestyle seen elsewhere in the Cnidarians. This represents a fundamental shift. Rather than building on the shared structures seen throughout the rest of the group, the body is reduced. Features that define other Cnidarians are simplified or lost. What remains is a highly specialized organism, adapted to life within a host. In some cases, this reduction is taken to an extreme. Certain members of this group are among the smallest animals ever discovered, composed of only a handful of cells, lacking many of the structures we would normally associate with animal life. And yet, despite this transformation, their origins are still visible. Within their cells are structures that resemble the stinging apparatus of other Cnidarians, modified but recognizable. These are not used for capturing prey in open water, but have been repurposed for anchoring within the host. This connection was not always recognized. For many years, these organisms were thought to belong to entirely different groups. It is only through more recent study that their place within the Cnidaria has been resolved. Even now, much about them remains unknown. Around 2,000 species have been formally described, but estimates suggest that tens of thousands may exist, many still undiscovered. In this lineage, the Cnidarian body plan is not expanded or elaborated. It is stripped back. Tentacles are lost. Mobility is lost. Even the distinction between polyp and medusa disappears. The organism becomes something else entirely, a parasite, living within the body of another. And yet, the defining feature remains. The cnidocyte, modified, reduced, but still present, links them to the same ancestry as corals, anemones, and jellyfish. This is the final pathway within the Cnidarians. From attached polyps to free-swimming medusae to microscopic parasites, the same fundamental biology has been reshaped again and again over hundreds of millions of years. A simple body plan, repeated, modified, transformed, and from it, an entire phylum emerges. At first glance, these animals can seem simple, lacking the structures we often associate with complexity. No centralized brain, no obvious hierarchy, and yet, from this foundation, an extraordinary range of forms has evolved. Coral reefs that reshape entire ecosystems, drifting jellyfish that move through the open oceans, parasites so reduced they barely resemble animals at all. The simplicity was never the limitation. It was the starting point. I hope you enjoyed this journey into the Cnidarians. If you're interested in the research behind this episode, you can find a document with image credits and sources in the description below. These videos take a great deal of time and research to produce. They will always remain free to watch. But if you would like to support the channel and gain access to new videos a little earlier, you can find a link to my Patreon below as well. If this video sparked your curiosity, don't let it stop here. Share it, talk about it, and keep learning. There's still so much to discover about the world's rare and remarkable species. And the more we understand, the better our chances of protecting them. Thanks for watching.